All right. So sorry about that. Computer had to update, but I think we are about ready to go. Thank you for those of you who filled out that lecture comprehension survey at the end of class last time and the study habits survey. I'll get back to you with those results after I have some time to dig into the data. Looks like the class is a little bit split on most common answers I saw were I get the sodium potassium pump, I still don't get membrane potential, or the exact opposite. So we'll touch base on both of those as they become relevant, and I'll keep an eye on those and we'll keep practicing. And if you have any specific questions for me, I'm happy to talk through them. Okay. But when we left off, right, what we were talking about was setting up our resting membrane potential for drugs. So membrane potential, we should hopefully remember or try to understand. A membrane potential is like something you could measure. It's a measured state of the situation that your cell is in. What you're measuring is like the voltage inside the cell versus outside of the cell. So that's due to all the ions inside the cell, all the ions outside of the cell, and the difference between them. So you could go in and measure a membrane potential of any cell at any time. When we say a resting membrane potential, we mean that neurons, our nerve cells, have a specific kind of steady state that they start at. So that specific steady state that they're starting at is a resting membrane potential of -70 millivolts. Right? That's just something we observe generally to be true. And we were talking about how that gets set up using our sodium potassium pump. Our sodium potassium pump is our first and most important example of primary active transport. Primary active transport because it uses ATP to pump ions against their gradients, right? Push compounds against their gradients, in this case ions, right? So we push 3 sodium out, we pull 2 potassium in every time the sodium potassium pump does something. So that means we end up changing concentrations of sodium inside the cell versus outside the cell, changing concentrations of potassium inside the cell versus outside the cell, and changing overall how many positive charges we have outside the cell versus how many positive charges we have inside the cell. So we're kind of depleting the inside of the cell of positive charges because we pull so many sodium out and only put some potassium back in. So we're talking about how that gets us to setting up this resting membrane potential of neurons and understanding what direction the forces are for our 2 most important ions when it comes to neurons, are sodium and potassium. All right. So what we end up developing is a situation where we have a chemical force for potassium going out of the cell, right? So that's our CDF for potassium, okay? So that chemical force for potassium is out of the cell, and the potassiums are the blue-purple ones, okay? That's our chemical driving force. So that's running down our potassium gradient, which is high inside the cell. The sodium potassium pump pulls potassium into the cell, low outside of the cell, because that's where we got that potassium from in the first place. We have the reverse chemical driving force on sodium. So our CDF is the opposite direction. So we jammed a lot of sodium outside the cell and emptied the inside of the cell of sodium. So we now have a chemical driving force on our little red sodium ions to move in. And when our cell is negative, which is what we see happening first as we're getting to the resting membrane potential, we know we're going to -70, right? So we're going to really focus on our negative side of the equation right now, right? So we're negative because we took 3 sodium out, replaced them with only 2 potassium inside, right? So that was 20% of the reason we have this -70 charge inside the cell. This is the direct action. And then we had a lot of movement of ions after that, kind of indirect effects of the movement of that pump, getting us all the way down to -70. But once we do have a negative charge inside the cell, so a negative membrane potential, this means our electrical force for sodium, which is a positive ion, is into the cell. And so is our electrical force for potassium, because it's also positive, and positives and negatives attract. Okay. So this means that we have a really big force for sodium to come into cells, into our neurons. So our electrochemical driving force on sodium is really big, moving into the cell. And our electrochemical driving force on potassium is weakly in the opposite direction, right? So this electrical force is kind of combating some of this chemical force. So on net, our total force for potassium is out of the cell, but it's a little bit smaller. Okay. So even if, actually I should say, especially if you're still having trouble with the electrochemical driving forces, important, it's helpful to understand how these things develop and how they move. But to keep you with us, you can start by just memorizing with neurons. Our sodium wants to come into the cell because that's its force, sodium coming into the cell. And our potassium in general wants to leave the cell because that's the direction for the force. So that's always going to be true at the resting membrane potential. And it's going to be true for our neurons like throughout most of their behavior, right? So even if you're still a little fuzzy on why, definitely memorize sodium wants to come in, potassium wants to go out. I'll get you at least part of the way there. Okay. So we've developed all these torches, and at a certain point, we start to develop a steady state that is dependent on the number of leak channels for sodium and potassium in our typical neuron. So we have more channels for potassium. So those are those three blue ones up top for our potassium. So we have potassium flowing out, more of it flowing out originally than we had sodium flowing in, which is why we were down near-ish potassium's equilibrium potential of -94. But as that force for potassium to leave the cell gets weaker and weaker and weaker because the cell is getting more and more negative charge pulling that potassium back in, we start to end up with a state where like our potassium is moving so slowly out of those potassium channels, and sodium wants to get into the cell so bad that we end up with kind of a 1-for-1 replacement. And that point is around -70 millivolts. So when the cell gets to -70 millivolts, this is when we're kind of stabilized. All right. So we're stabilizing here, and to make sure that we don't run out of sodium and potassium in the appropriate places, that sodium potassium pump keeps going. So keeping those concentration gradients how they were, because otherwise eventually, you know, like we'd empty the cell of all the potassium potentially, or like we take all the sodium and bring it in, right? We keep this pump going to maintain that kind of status quo here. Okay. The questions about this at this point. Our basic thing that we want to know is that sodium wants to come into cells, potassium wants to leave cells. Potassium has more channels open for it, so it's kind of winning in the fight between sodium and potassium. The sodium potassium pump is kind of remotely driving. All right, so that brings us back to this picture we saw before, right? We have our resting membrane potential here down near our equilibrium potential for potassium, which is -94, our equilibrium potential for sodium, which is +60, right? And so we have sodium wanting to flood into the cell to raise that membrane potential up, and potassium lightly wanting to escape the cell, because if you remove a positive charge, you become more and more negative. So When we have a membrane potential that is not at the equilibrium potential for an ion, this means we have an electrochemical driving force, not zero. That force moves the ion in the direction that favors being at equilibrium. And the farther we are from that membrane potential, the farther that is from the equilibrium potential, the stronger our force is. Cool. Saying this stuff again, right? So when we are at our resting potential in the, okay, I am emerging. Sorry, I'm just suddenly noticing how many times I repeat myself. Okay. Okay, so resting membrane potential of -70, right? We can notice that our equilibrium potential for potassium is about 24 millivolts away, right? So we need to go down by 24 millivolts to get to that equilibrium potential. The situation that we have set up, which we know because we just looked at the cell, we drew it out, we drew those concentrations for our potassium, sodium, we drew all those positives and negatives. We know that we have a chemical force going out of the cell. We have an electrical force and attraction to that negative charge into the cell. But overall, that chemical force is winning. That concentration difference is really big. Which is why we have this moderate force for our potassium to still leave the cell, even though that electrical force is kind of still pulling it inwards. So the opposite situation comes for the sodium, right? So we have a really big difference between our equilibrium potential for sodium and our existing membrane potential, right? So we have a difference of 130 millivolts. So this is a really strong force, right? We're really far away from like our desired equilibrium point, right? So that we have a really strong force for sodium to move, okay? And in part, at the resting membrane potential, this is because our electrical and chemical forces are both pointing the same direction, so they have this additive effect. Right? Nothing's slowing anything down. The thing is, we don't get to our equilibrium, right? We don't get towards sodium's equilibrium point yet because we have really low permeability to sodium. So sodium wants to move, but it can't because there aren't enough channels. They're full. That's how we're starting it. Keep that membrane potential going, that resting membrane potential. The sodium potassium pump is acting, keeping sodium and potassium gradients stable. We've got a couple of leak channels here. Only one in this picture, very few in reality for sodium, more of them for potassium, but relative to the processes we'll talk about as we go through the action potential and the communication of neurons, these are both pretty small amounts of sodium and potassium moving. And we know we have these forces, right? So we have a high force for sodium, right? Wanting really bad to come into the cell, it just can't. The reason we don't have much potassium escaping, even though we have these open channels, is that force for potassium to move is kind of weak. So you can see these things move around. The textbook is amazing at making video.
The human brain alone contains about 100 billion nerve cells, all neurons. A neuron, like every other cell, has positively and negatively charged ions inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So that's all we were really trying to understand in this first bit. And that is our setup for moving towards understanding how a membrane potential of a neuron is going to change over time in a way that lets us send those little shocks, like the voltage, the shock, right? Like this is the electrical activity of the neuron. And we're going to send these little shocks down the body and down the axon, ultimately, of our neuron so that we can talk to other cells, right? So what we're going to understand at the end of this unit, right, is how we go from a starting point of negative 70. So our resting membrane potential is going to be at negative 70. This is our resting membrane potential. So we're going to start there. We're going to end there, right? So negative 70. Right? But in the in-between, when we're sending a signal, we're going to see that the membrane potential of that cell shoots up to positive 30, right? So we're going to have this wave of positive charge flowing into the cell, mostly because sodium is coming in, right? And then to get back to the resting potential, we're going to see that that membrane potential has to fall back down. So we're going to see a flood of positive charges out of the cell at this point, bringing us back down more negative until we stabilize back at our resting state, right? So we're going to see this spike of charges when we're looking at that membrane potential over time. And that's what's ultimately going to trigger the release of things like neurotransmitters, talking to the next nerve cell in a line. So that's our key thing that we're trying to understand, is like why we have membrane potentials shift. So we are also going to be talking about where in a neuron we are at a given time when we're talking about these different types of channels in these different charges. So you have this picture on your slides as well, and we're going to go through and kind of match them up so that we have like the terminology of the different parts of like a super simple neuron. There are lots of different types of neurons. They don't all look like this. But this is going to be our simple, easy, basic, most common neuron shape. So we're going to go with this one as we try to understand what's going on, creating these action potentials and creating this communication. Okay. So when we're talking about a neuron, we're talking about like its main part, its like main body. Right? We're talking about the part where like all its organelles are and stuff. Right? If we're drawing a circular cell, like we were just drawing those circle neurons before, that's our soma, right? So the soma is the central inner kind of like part of our neuron. So that's the body of the neuron here, the soma. Okay. Now on either side of this, right, like our neurons are meant to both receive signals and send signals out. Okay. So we're going to think first about the incoming signals. So these little green arrows are showing you direction of charges, basically, direction of signals. All right, so these little hairs off the soma here with arrows coming in, that's showing you signals coming in down our dendrites. The dendrites are the part of the neuron that bring a signal into this soma, into this body of the neuron. So when we talk about graded potentials, which is going to be our next topic before we get into our big action potentials, we're going to be thinking about those happening on our dendrites and maybe these little regions here on the soma between the dendrites as we have signals coming in, things happening here, kind of head end of our neuron. The rest of the cell is focused on sending signals out, right? Our nerves are sending signals somewhere. That's kind of their job. Right? So the main highway that they travel is the axon. But before we get there, we're going to see that we have this little pinching off area here. So this little triangle I'm drawing, this is your axon hillock, right? So that's the axon hillock. That little triangle, that little pinching off point leading us into the axon is the axon hillock. And that's going to be important because that's like the on-ramp to the axon, right? Our axon hillock is our on-ramp. This is where our action potential is going to start. And then as we pass our signal down, what we're really going to be focusing on when we're passing a signal, so we're going to have a series of membrane potential changes going down, 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 this structure called the axon. So the axon is this long skinny part. So that's our axon. So that's how we're sending the signal down the length of our neuron, down the length of this axon. In order to actually send a signal fully to another cell, we're going to want to communicate with its dendrites or maybe its soma, right? Occasionally other places, we'll talk about it if they're relevant. But we're going to do that by releasing chemicals, right? So we are carrying within the structure of the neuron, we're carrying that electrical signal, that membrane potential change. But our end goal is usually going to be to release a neurotransmitter. And that's happening at these little buttons at the end. Those are your axon terminals. So terminal, like end, that's at the end of the axon. Those are our axon terminals. So as we go through our next packet and talk about the action potential, how it develops, how it travels, how it gets passed on, we're first going to be thinking about what's happening like here at our head of the cell, the soma, and the dendrites as we're receiving signals, as things are affecting the neuron. And then we're going to be thinking about, okay, how do we pass that along? And really, we won't start to think too much about the axon terminals until we start to think about synapses, the neurotransmitters, and communication. So questions about this before we move into that next set of topics? So you will want to learn these parts because we're going to be thinking about where different types of channels are, where different membrane potentials are collecting, where we're seeing changes, but hopefully that part's relatively straightforward. I do say that as an anatomist, I love naming body parts, so that's not your jam. Sorry, we do still need to know the parts. Okay. So we're going to start by thinking about types of electrical. Okay. So when we're talking about electrical, what we're thinking about really, like from a physiology basis, changes to this membrane potential. Okay. We're going to have different amounts of change to our membrane potential that serve different purposes. Okay. So our membrane potential, right, is just our measurement of what is the cell at right now. So like what is this region of the cell at right now? We're not going to go more specific. Right? Our graded potentials and action potentials are naming our big picture changes or specific types of changes. Okay. So what a graded potential is, and so this figure A is our graded potential. So graded potential. So we're going to have a little membrane potential. So we'd still start at like -70. We see little spikes, right, in response to stimuli. So these pink arrows are something happened. Okay, so that's our stimulus. Okay, so you see something happened. We had a little graded potential. Something else happened. Another little graded potential. We can go up. We can see membrane potential go up. We can see it rise. We can also see it fall. So this little dip, also just a graded potential. So our graded potentials are these small changes to the membrane potential. And they're going to dissipate over time. And they're really going to only communicate over small distances. So graded potential is going to affect like neighboring parts of that neuron. But a graded potential isn't going to travel the full length of a neuron and into another cell, right? So the graded potentials are going to be kind of like small local changes. And we'll talk about that. When we think about our action potentials, so an action potential is always going to look like this exact curve, pretty much, and in that exact order. Okay. So an action potential is big, and it's a specific amount of big. It peaks at +30. What you're going to see with our action potential is that this is the membrane potential change that your neuron uses to flood the entire neuron kind of in like a wave form or kind of like hitting a stack of dominoes, go all the way down the axon and get to the end of the cell, to the axon terminal, so that we can send a signal to the next cell. So it's large, and the action potential is going to communicate over long distances. It's going to go all the way down the axon, which is that long skinny part of the cell. Okay? And it's going to be very consistent in what it looks like. So here they're showing you actually a tiny little graded potential first here. So we have a resting membrane potential. Here you see a little graded potential that didn't do anything, right? And we went back to rest. But here you see we went up, and when we hit this membrane potential of -55, that's what we think of as a threshold. And that kicks off this whole spike and dip before we go back to rest. That spike is going to go to +30. And that curve, that spike and dip, and then back to rest, that is our action. And we'll talk about how and why, and you'll see it again a bunch of times, but that's what we're talking about. So I want to remember when we're thinking about sodium, which is going to be key to understanding some of these things, which of these be true or untrue? So pick which is untrue of sodium when we have a membrane potential that's at rest. So we're at -70. Check if each of these things are true. If one's not, pick it. They're all true, pick the last one. Okay, let's take a look. Cool. Okay, so they are all true, right? So the situation that we set up last time, right, or in the last packet, is we've got our cell. We're going to think about this now as our soma of our neuron, right? So we have a force for sodium to come in. So our electrochemical driving force is strong on sodium. So that's our electrochemical driving force. Pointed in. We're thinking about sodium right now. Okay, cool. But we only have a few channels. So not much sodium leaks. So we have only a very slow leak because we only have a few channels. for sodium available. Versus if we were talking about potassium, our situation would be different because we have a lot of, relatively compared to sodium, a lot more potassium channels, but its whole force situation is different. It wants to leave cells and the force isn't that large. Okay. So for sodium, the reason we don't have much moving is that we don't have very many channels, even though we have this really strong force. So let's think about our graded. Okay. So graded potential is going to be one of those small changes to the membrane potential. Okay. And it's going to be happening in a location. So it's going to happen when we have some type of a signal or stimulus come in part of our cell. So in this picture, it's probably showing like part of the soma, but you might think it's coming in down a dendrite too. Okay. And it's going to create a little bit of a change right where that stimulus struck us. Right. So we're going to see a stimulus. So here we see a stimulus creating a little change in that membrane potential. So the thing that is true about graded potentials, but won't be true about action potentials, is that stimuli of different sizes create graded potentials of different sizes, right? So here we had a small stimulus at X with a small little graded potential. Here we have a slightly stronger stimulus. So we had a slightly stronger change on the membrane potential. So we have a difference in magnitude between different graded potentials, which is why they're called graded potentials. Right? So they have this change in how big they are depending on what type of signal, how much of that signal came. So these stimuli coming in, right, we're getting like little zaps from neighboring neurons, or maybe we're touching something, right? But these stimuli are happening at a location, right? So here you see that stimulus coming in, hitting some specific location on our cell here, on our neuron. All right. So that location is where you're going to have the biggest effect of that graded potential. So we have a big change in the membrane potential if you were to measure the voltage across this part of the membrane over here right now. Right, so here we see that recording of the change to the membrane potential. So that little graded potential here at the stimulus site. But in order to have an effect on the rest of the cell, right, you can think basically about these charges kind of diffusing across the cell, spreading out, right? So that stimulus and that graded potential are going to affect some other parts of the cell, but they get weaker and weaker and weaker and weaker as they get farther away. So if we were to measure the change to the membrane potential at the axon hillock, right? So it's that pinching off point of the neuron. So in this picture, it's on the opposite side from the stimulus. You can see there's still a change to the membrane potential, but it's weaker than if we measure it at the stimulus site. So that's what we mean when we say that our graded potentials are decremental. So decremental, they decrease or decay if you're measuring at a distance far from that graded potential's origin, from that stimulus site. And that process of spreading out, right, that process happens through something called electrotonic conduction. Electrotonic conduction is just the spread of the charges. Okay. So we don't need to know too many details about the electrotonic conduction, but we really want to know that we have an effect on farther parts of the cell to some respect, but we're getting weaker and weaker and weaker, decreasing the effect as we get farther from that stimulus. Right. The reason we care about our graded potential in large part is your neuron is sitting there trying to figure out whether it's supposed to signal the next cell or not, right? Like, so we're sitting here at our resting membrane potential, trying to decide, do I fire off an action potential now or do I not, right? And so what's going to trigger the release of an action potential is how many signals, how many stimuli you have coming in from neighboring cells, from the environment, whatever, creating graded potentials. Your neuron is basically adding those all together and figuring out, like, okay, is this a big enough of a change that I should send an action potential and tell the next cell in line. So these graded potentials, these little small changes that we've been tracking, are going to determine whether you send an action potential down the axon all the way to the end. But it's not the case that every time there's a graded potential, there will be an action potential. It's a decision point. It's a yes or no kind of situation. And how we figure it out is whether we hit this specific threshold. So we're going to have a threshold value of around -55. Oops. Right. So we're going to have a threshold value around -55, but we'll talk about it. Okay. And if we have changes to the membrane potential, the axon cell outside, like get us to that threshold, right? So if you end up with graded potentials adding up enough that you hit that threshold, then you fire off an action potential. If you don't, those graded potentials are just kind of like slowly fade away. So that's what we're trying to figure out when we're trying to figure out, are we sending an action potential? So the formal way we would describe that threshold is this is the level of depolarization necessary to elicit an action potential. So it's depolarization because we're at -70. This is like -55. All right, so 0 would be up here somewhere, right? So depolarizing the cell, right? When we say we're depolarizing, it's not polarized if you're at 0, right? So 0 is like what we're comparing to. That's no polarity to the cell, no plus or minus. So if you go from -70 up to -55, you're now closer to 0, you're less polarized. So that direction, right, that direction of change towards 0 here is depolarization. So the threshold is how much depolarization, so how far up do you have to go? How close do you have to get to 0 before we send off an action potential? How do we trigger that? That's our threshold. Okay. So this means that when we see depolarization, so when we see changes towards 0, right, and towards that threshold really is what we're thinking about. Those depolarizations we're going to think of as being excitatory. Excitatory, like we're making the neuron excited, it's getting ready to fire off an action, right? A cell is excited if it does something. So those depolarizations, we're getting a slightly less negative membrane potential, makes it more likely that our neuron will do something. So excitatory and depolarization go hand in hand. Not all our graded potentials are in that direction though, right? So we can also have graded potentials that make the cell even more negative, right? So that go down, right? So if you go from like -70 to -80, right? You've gotten farther away from 0 and farther away from your threshold. Okay. Cool. So this is making it even more polarized. It's even farther away from 0. It's even more negative. So that graded potential we refer to as hyperpolarizing. So when we have hyperpolarizing graded potentials, they tend to be inhibitory. So inhibitory, you're inhibited, you don't do something, right? So we've gotten even farther away from our threshold necessary to send off an action potential. So we've made it even less likely that your neuron's going to do something, less likely that your neuron's going to send a signal down the line. Questions about this before I ask you all? We don't really use that term, but you might think about it that way. When we use these terms, we're usually describing the action potential specifically, which always has that specific form. But yeah. So So that could be a good made-up term, but in practice, we're just not going to observe it that way because of where we are starting at. Okay, cool. All right. So we're going to relate this to some ions, okay? Okay, so say we have a positive ion, a cation ion. We're going to think about any of them right now, just so your generic positive ion. Okay. So if I'm observing a depolarization, right, or an excitatory potential, an excitatory reaction, what direction did I move my ion, right? So we had a positive ion, sodium or potassium, move into or out of the cell to change that membrane potential, right? Which of these is going to be responsible for causing the depolarization and the excitation? Okay, we got a 50-50 here. All right, cool. Yeah, so the depolarizations are going to be caused by positive ions moving into the cell, right? So when we have that positive charge come in to our cell, and if we were at -70 and we add a positive charge into the cell, well, maybe we're at -69 or something, right? Okay. So that's that depolarizing direction, that up towards zero direction is going to be caused by cations moving into the cell. Okay. Which means that we're really thinking about sodium moving into cells and causing depolarizations. Okay. So I'm going to send you these questions to do later for practice, because we're going to keep moving a little bit and just mention that we can sum these graded potentials together. So we'll start with those questions next time and go through. Okay. So we've got these graded potentials. There are changes to the membrane potential because we either had positive ions flowing in, positive ions flowing out, usually our sodium and potassium. Okay. And we're trying to get to our threshold, right? So we're trying to get to this threshold of -55, right? This is when we get to this threshold that we kick off our action potential, which is that spike in that dip. Okay. Now, we don't need to wait for just one enormous stimulus in order to get to that threshold, because we see that our graded potentials can add together. So you can have like 100 little pokes at once. They all poke you in the depolarizing direction, in that excitatory direction, right? You can kind of add them together and they will get you closer to that threshold and send off an action potential. And there are two ways basically that you can get graded potentials adding together. So fundamentally, you can have them add up over time, or you can have them add up over space. Our temporal summation, what that's saying, so if you have a stimulus, right, and it keeps going, right? So it's not just one poke letting a sodium ion into the cell, it's poke, 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 poke over and over again. They would add together over time, right? Like that frequency of stimulus after stimulus, repeated close together in time, can result in adding up to a larger graded potential that hits threshold and kicks off an action potential. Or the other thing that can happen is you can have two signals coming in from two neighboring cells, or three neighboring cells, or four neighboring cells. And if they all poke you at once, and they all happen to be depolarizing graded potentials, depolarizing excitatory stimuli, then you add them together. They can also kick off your action potential. If you add together a a depolarizing stimulus and a hyperpolarizing one, you can end up getting like a flat line. They can cancel each other out. But all we're talking about is adding together over time or adding together over space, just trying to figure out, do we get to this threshold to kick off our action potential? Okay, so we'll leave there for today. Next time, we will start with these couple questions to make sure that we understand the depolarization, hyperpolarization, and the ions. And then we'll do some examples on those great potentials. And if you can go ahead and let me know which parts of today's content you feel shakiest on versus most confident about, that would be helpful. I'll try to write those into our questions. This is also serving as a record for you when you go to study at the end of the unit. Oh, I was super confused on day one when we talked about sodium potassium pump. Hopefully by that point you won't be confused. If you are, you'll know it. Yeah.
So it was directly off one of the slides from the first slideshow.
From today or from?
From today.
Yesterday? Okay, cool. Do you know which one it was?
And maybe it was a review yesterday. It said something about like why like the difference between like sodium and like the like baseline.
This one? You can click there. Or you can ask me what the question was.
This one.
Oh, okay, cool. Mhm.
Why would it be less negative than because isn't
Mhm,
that
mhm, mhm.
more negative?
Ask me the question.
No, this is something that would help.
Mhm.